Hello, this is my final project for Precision Medicine Program, Applications of Transcriptomics and Machine Learning to Post-mortem Interval Estimation, a preliminary study. Let me introduce myself. I am an assistant teaching professor and graduate program director of the Professional Science Masters in Forensic Science at Florida International University in Miami, Florida. I am a forensic scientist with a background in biomedical science. My projects apply transcriptomics, proteomics, and epigenetics to forensic science issues, like HR death estimation or determination of post-mortem interval, like this project. Apart from the Precision Medicine program, I completed 2 other programs, OmicsLogic data and Transcriptomics in biotech. And now let me introduce the project. There are several methodologies to estimate the post-mortem interval, PMI, or time since death based on changes that undergoes after death. However, these methods are imprecise due to the composition process being affected by several factors. Current trends attempt to estimate the PMI in a quantifiable manner based on chemical, biological, and microbial changes, leading to the fields of thanatochemistry, thanatobiology, and thanatomicrobiome. So, based on transcriptomic data from mice tissues, the aim of this study is correlating gene expression changes with PMI, leading to estimate in a quantifiable manner time since death. For this, I used transcriptomics data from a previous publication from mice neuroretina and retinal pigment epithelium, retrieving from 0 to 24 hours after death. Data were analyzed through the bioplatform applying unsupervised and differential expression analysis. Additionally, correlation coefficients and unit and multivariate regression analysis were performed in significant genes. First, neuroretina and RPE had differential gene expression, and using exploratory data analysis, in RPE, PCA show a clear separation between the early PMI groups and the late PMI groups. Also, applying clustering, K-means indicated this separation. In retina, PCA and K-means results were variable. However, both tissues displayed differential expression of genes through the PMI, some of them with strong significant correlations. Based on this data, gene expression could be quantifiable correlated with the PMI, although this gene correlation depends on the tissue and signaling pathways activated. And as conclusions, this study provides a quantitative method to estimate the post-mortem interval based on transcriptomics data. Future research may be able to expand the results applying in human tissues and extending time since death estimates. And let me introduce a little bit the whole post-mortem interval problem. So post-mortem interval is defined, post-mortem interval PMI or time since death is defined as the time from when the person died to the time when the corpse is discovered or the corpse is sent to the lab. There are classical methods to determine the post-mortem interval, and these are based on changes that a corpse undergoes after death. Also, forensic entomology is based on insect activity, the colonization of different species. However, this method cannot quantifiably determine the post-mortem interval and also are affected by different environmental factors. For that reason, new methodologies are trying to estimate the PMI in a quantifiable manner, taking into account these influencing factors, like temperature and humidity. The first developed method is called thanatochemistry and is based on chemical changes. For example, the analysis of volatile fatty acids. Recent trends are based on molecular biology, thanatobiology, and microbial colonization, thanatomicrobiome. For this project, I focus on thanatobiology methods. Thanatobiology methods have been recently developed, and these new trends have been analyzed at different levels: DNA degradation, RNA degradation differential expression, and protein degradation expression. These methodologies could improve the PMI estimates as next-generation sequencing technologies allow to analyze several samples and genes at the same time, quantifying expression levels. This could lead to better and accurate PMI estimates. So, based on transcriptomics data from mice neuroretina and retinal pigment epithelium, the aim of this project is to study the differential expression of genes through the PMI to find a correlation between this expression and the time since death. As material and methods, I use processed transcriptomics data from a previous publication, this one with this geoaccession number. Briefly, the authors used C57BL6J mice sacrificed at 30 days of age, and they extract neuroretina from now on, retina and retinal pigment epithelium RPE from now on at different times after death: 0, 15 minutes, 30 minutes, 45 minutes, 1 hour, 3 hours, 6 hours, 12 hours, and 24 hours, and 4 mice per PMI. This data was analyzed using first exploratory data analysis using principal component analysis. But first, I performed quantile normalization and then the PCA. And also the data was clustered using k-means. And finally, the differential expression of the genes was analyzed using the DISSEQ2 algorithm. DISSEQ2 is a method for differential analysis of count data that uses a shrinkage estimation for dispersions and for changes to improve stability and interpretability of estimates. This enables a more quantitative analysis focused on the strengths rather than the mere presence of differential expression. And this is really useful for lower expressed genes. In that way, we can take into account these genes in the whole differential expression analysis. After the differential expression analysis, I perform correlations. And for data follow a normal distribution, I use Pearson correlation. Non-normal data, I use raw Spearman. And finally, I perform uni and multivariate regression analysis to create formulas to determine the PMI. In terms of results, first, I decided to analyze if retina and RPE have differential expression of the genes. So as you could see on the picture of the eye, retina and RPE are closed. So it was good to know if they have different expression, and in this case, treated them as different tissues, or if they have the same expression, the same type of expression. And for that reason, I used the time 0 to analyze this. And I applied the DISSEQ2 algorithm. The DISSEQ2 algorithm gave me two plots. MA plot, which represents the log-fold change, log of the ratio of level counts for each gene between the two samples against the log average, the average of level counts for each gene across the two samples. And the volcano plot. The volcano plot is a scatter plot of the negative logarithm transform p-values from the gene-specific test against log-fold chains. And as you can see, volcano plot gave me also the differentially expressed genes. As you can see from the red dots here and the green dots here, effectively, retina and RPE have different gene expression. For that reason, we have to treat them as they are two independent tissues. And the first thing I did was to analyze data using exploratory analysis, using principal component analysis or PCA. And as you can see here, although first I applied quantile normalization. As you can see here, in retina, there is not clear group separation. However, the two components explain 82% of the variance. In contrast, in RPE, we can see a separation between later PMIs, 12 to 24 hours, this one, and early PMIs, 0 to 6 hours. And the two PCA components explain 74% of the variance. So based on this exploratory data and the PCA results, I decided to see if the samples or the PMIs can be clustered. And I used k-means, and I got the same results as with the PCA. As you can see, in Retina, there is no clear separation of clusters. However, in RPE, I got the same results as in PCA. Later, PMI samples, 12 to 24 hours, clustered together. Early PMI samples from 0 to 1 hour, clustered together. And there was more variability on intermediate PMIs, 3 to 6 hours. I think in Retina, the problem was the variability itself of the sample. So for that reason, I couldn't see this separation. And now my next step was to analyze the differential expression of the genes. And I started with Retina. And I'm going to show you first the MA plot. And here you can see the analysis respect to a previous time point. I did also the analysis of each time point respect to 0, like this one. So for example, 0 versus 30 minutes, 0 versus 45 minutes. And I got the same results as the previous publication. Here I show you the analysis respect to previous time point to show you the tendency of the data. And as you can see, again, the red dots represent the significant differential expressed genes from 0 to 15 minutes. We can see there is a differential expressed genes, not so much from 15 to 60 minutes. However, later PMIs and particularly the last hours of death show the highest differential expression. I got the same result using volcano plot. And I show you this because I use volcano plot for the next step, the correlation analysis. And volcano plot points or marks the differential expressed genes. Again, in green, you can see the differentially expressed genes. And I also did from 0 to different time points, but here I show only the respect to the previous time point. And as you can see here, from 0 to 15 minutes, there is a differentially expressed genes, not so much in this case from 15 to 45 minutes. And then when I increase the PMIs, you can see the highest differential expression occurs in the last hours. Based on this volcano plot, I select these genes to do a correlation analysis. In bold, you have the significant correlation analysis. Significant correlation means that with the time, there is an increase or decrease in the gene expression. So for example, positive correlation means that there is an increase in the gene expression. Negative correlation, there is a decrease in the gene expression. And also, the strongest correlation is 1, the value 1. And here, for example, you have an example of a strong correlation. And based on this correlation, I graph the genes that were selected for the posterior regression analysis. And I got the strongest significant univariate regression using the SIR61 gene. And remember, it's a positive also correlation with an R of 0.913. And in multivariate regression, I got the strongest one with SIR61, RIC, and LF2. And RIC and LF are negative correlated. So here you have the function of the genes TIR61 is involved in inflammation and tissue reparation, which makes sense with the death event. Also, RIG and ELAF are involved in transcription regulation. So TIR61 is upregulated with the PMI. In contrast, RIG and ELAF2 are downregulated, or maybe their RNAs are degraded. So I'm not sure about this. It could be downregulation or the degradation of the RNAs. What is clear is that TIR61 is upregulated. Now I'm going to switch to RPE. And the same, this one I plot respect to the previous time point, but I did respect to 0 respect to any time point, and I got the same results as the previous publication. And here the tendency is similar as in Retina. In the first, so from 0 to 15 minutes, you can see differentially expressed genes, not so much from 15 to 60 minutes, but this increase in the later PMIs. And here is earlier at 6 hours, 6 versus 12 hours, we can see a high differentially expressed genes. And also the highest is from 12 to 24 hours. The same results using volcano plot. Here you can see more differentially expressed genes from 0 to 15 minutes. Also in the previous from 15 to 16 minutes. And then again, the highest differential expression is in the last hours. And again, based on volcano plot, I choose these genes for the correlations. You can see here involved again the significant correlations, and here you can see the majority of the genes were downregulated, or maybe the RNA is degraded. And here I plot the ones that were selected in the regression models. The strongest significant univariate regression was with ATL3, with an R of 0.854. And for the strongest significant multivariate regression was with ATL3 and PIC5, ATL and POT1B. And with Rs of 0.985 and 0.938. ATL3 is involved in axonal maintenance, PIC5 is involved in membrane homeostasis, and POT1B involved in telomere protection. So remember from here, POT1B was the only one with positive correlation, means that POT1B was upregulated with the PMI. So if it's involved in telomere protection, makes sense that because of the death event, try to protect, is activated to protect the telomeres. The other two, ATL3 and PIC5, are downregulated, or maybe the RNAs were degraded. So. And also this SEC2 pipeline gave me different affective pathways. It will be interesting to analyze these differentially expressed genes in each pathway and try to create regression models to determine the PMI. So as conclusions, this study demonstrated a differential gene expression through the PMI in mice retina and RPE. This differential gene expression depends on the tissue. And this differential gene expression was higher in later PMIs, 12 to 24 hours. Some of the differentially expressed genes show a strong significant correlation with the PMI. The regression formulas were created to estimate the PMI based on significant correlated genes. In order to accurately determine the PMI, it is required to take into account all differentially significant expressed genes and, as a result, a more complex statistical models. Future studies will be able to expand the results, applying in human tissues and extending the PMI estimates. Thank you very much.